Hey everyone, and welcome to this explainer. Today we're doing something honestly pretty incredible. We're taking those dense, kind of intimidating Cambridge Biology 9700 syllabus requirements, and we're transforming them into a real-world biological mystery. And the best part, we're going to solve it together using a totally visual, step-by-step strategy. So if you want to know exactly what those examiners are looking for, well, you're absolutely in the right place. Let's just dive right into it.
So we start with our central mystery. Have you ever wondered why some candidates can perfectly memorize complex concepts, like, say, the Apoplast pathway, but then they just completely fail the exam question? Well, according to the principal examiner reports, it's rarely a lack of biological knowledge. It's actually a failure of execution.
Section 1, Unlocking the Examiner's Playbook. The secret to crossing those elite grade boundaries isn't just about cramming facts. It's about mastering a highly specific framework that examiners literally hunt for in every top band answer. Okay, so how do we actually solve this? We use the four-part method mark strategy. Step 1, you deconstruct and extract the given context. Step 2, build that scientific chain. Step 3, execute with crystal clear notation. And Step 4, refine it all with precise examiner wording. This system is a game changer because it ensures you secure points for your logical process even if your final calculation ends up being a little off. Let's see this framework in action right here on screen with the mitotic cell cycle. A massive 15% of paper 2 marks are often hiding in this topic alone. Notice that warning zone on the right? Flawed answers tend to confuse cause and effect, right? But top band answers, they actively use comparative connectives and highly specific syllabus terminology to absolutely lock in those marks. Section 2, Mastering Biological Transport Systems. Now, we're going to apply our new detective framework to some of the absolute most complex mechanisms in the entire syllabus. Take a look at the difference here. On the left, you've got a flawed, generic student answer that basically just spits out textbook definitions about water movement. But on the right, the top band answer directly applies the Apoplast pathway to the very specific context given in the question, complete with exact units. The big takeaway here, generic answers are traps, guys. Context is king. Taking over the screen now is the A-star strategy for the mass flow hypothesis in phloem. Notice how the elite candidate doesn't just list facts, no. They extract specific data from the stimulus to actually prove the hydrostatic pressure gradient. They're connecting the evidence to the biological concept using clear causal relationships. And that is exactly how you show the examiner you truly understand the mechanics. All right, moving on to this epic unseen mechanism that is literally taking place inside you right now. It's the ultimate real-world mystery. How on earth does your blood know exactly where to release oxygen during transport and gas exchange? Avoiding vague generalizations is absolutely critical here. A flawed answer will just say something like, blood transports these gases, and completely forget the units. To hit the top band, you've got to cite specific evidence about oxygen and carbon dioxide transport. You have to state the principle, pull exact data straight from the provided tables, and back it up with scientific reasoning. That is literally the only way to cross those grade boundaries. But hey, before we dive deep into the data, let's clearly define the bore shift so we know exactly what biological principle we're extracting evidence for. Put simply, it's the decrease in hemoglobin's affinity for oxygen in response to an increase in the partial pressure of carbon dioxide. Now, this brilliantly illustrates those mechanics. Looking at this high-contrast graph on screen, you can visually see the shift happening. Top mark answers have to explicitly state how the oxygen dissociation curve shifts to the right as carbon dioxide concentration increases. And they do this by pulling specific saturation percentages directly from the axes to prove their point. Section 3, Decoding the Immune Response. Let's shift our micro-lens to your frontline defenders and see exactly how examiner traps catch students off guard in this high-stakes topic. So when describing the mode of action for phagocytes, marks are so frequently lost just through simple terminology mismatches. Check out those red pennilets right in the center of the slide. Students mix up the specific roles of macrophages versus neutrophils all the time. You really have to define the precise biological principle before designing your answer to make absolutely sure you're matching the command word to the correct cellular defender. Looking at long-term immunity here, the examiner reports reveal a pretty crucial strategy. When you're comparing the secondary immune response against the primary one, you must use comparative connectives. We're talking words like similarly to highlight shared features and whereas to contrast the speed and scale of the responses.
If you just write down memory cells do stuff, you are looking at a big red 0. Section 4. Acing Data and Statistics. Look, this comes down to a harsh truth. Biological theory means absolutely nothing on exam day if you can't interpret the complex data and graphs representing it. To really master those higher-order evaluation questions, you have to systematically select the correct statistical framework for the evidence they give you. It is not a guessing game, guys. It's a decision matrix. And this matrix beautifully maps out exactly when to use which test. You use a t-test for comparing means of continuous data from two groups. You use chi-squared for categorical differences, comparing observed versus expected frequencies. And you use correlation tests for relationships between variables. Knowing this table back to front is literally like having the exam cheat code. We can also apply this exact same logical sequence to genetics. Notice how this framework takes really complex monohybrid and dihybrid Punnett square crosses and turns them into a simple, step-by-step deduction. Identify the core concept, extract your data, apply connective reasoning, and finish strong with syllabus-defined terminology. It takes all the panic right out of genetics questions. Section 5, the final exam takeaway. Let's go ahead and lock in everything we've uncovered in our masterclass today so you can walk into that biology exam with total confidence. Based on the principal examiner reports, these are your ultimate rules to live by. Omitting units, using vague terminology, and failing to relate your answers to the specific context are by far the fastest ways to lose really easy marks. Seriously, don't just write a textbook definition. You need to write a specific solution to the biological mystery they put in front of you. I really want you to burn this quote from the examiner alerts right into your memory. Students lose marks by not showing working. Your logical process, your step-by-step reasoning is just as valuable as your final biological conclusion. Don't skip steps. Secure those method marks. So now that you can literally see the hidden mechanisms behind the questions, the choice is entirely yours. I challenge you to apply this 4-step visual framework to your very next past paper. Are you going to fall for the generic answer trap, or are you going to command the mark scheme? Thanks so much for joining this explainer, and remember, keep questioning the world around you.